Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 13, which is viruses. And we'll want to talk about viruses and their role in cancer. So cancer, when I say cancer, I'm thinking of like a tumor cell, right? A cancer is a group of cells. These are your body cells that are replicating when they shouldn't be. Right? That's when you get a growth of cells. You get a tumor. So if these cells are replicating when they shouldn't be, they form this mass. And as that mass gets larger, it can interfere with other body systems. It can break off and move through the body and land somewhere else and form a new mass of cells there. So this is what we know of as the disease cancer. There's lots of different types of cancers from that. All right, so why are these cells replicating? Epithelial cells are supposed to replicate. Your hair cells are supposed to replicate. Your immune system. There's some of our cells that are supposed to keep replicating. Other cells, no. Our heart cell, it's supposed to make a heart and then stop. It's supposed to make a kidney and then stop replicating. How does it know to stop replicating? It's in the instruction manual. There are DNA, there are genes in the genome that are responsible for shutting down replication. It gets to a certain point and those genes stop replication. The names of those genes or those categories of genes are called Proto-Oncogenes, and they're involved in regulating DNA replication. All right, so if you have normal genes, proto-oncogenes, your cells will divide until they make a certain point and then they stop and they don't replicate anymore. Okay, but over time, we get some mutations, right? Mutations, you get spontaneous mutations, but even about that, every time we're exposed to mutagens, chemicals, X-rays, UV light, over the course of our life, we're exposed to these things and they can make little point mutations throughout our cells in our body. You've got a lot of redundancy, and most of the time, remember, the genome is big, and most of those things, some of those things are not genes, not recipes, but shopping lists and instruction, blender things, whatever. It doesn't always genes. So we get these point mutations all over, and usually we can handle a fair amount of them. When we get too many, or if we get them in the wrong spots, if we get them in those proto-oncogenes, then replication turns back on. And you get a mass of cells in your kidney that starts growing when it shouldn't be, and now you have a cancer that started in your kidneys. Okay, so that's defining proto-oncogenes. Proto-oncogenes are genes that play a role in cell division. Most of the time they are off. They get to a point they turn off, and then we don't have unregulated division of our cells. But we build up these mutations, and at some point, if chances happen this way, if we get enough of these mutations and over time, we can hit these proto-oncogenes and then they stop working. And then those cell replication instructions turn back on. Then we get a tumor. Okay, so what does this have to do with viruses? Okay, we have found that viruses can increase that mutation rate. So if I'm exposed to a virus at some point in my life, in particular viruses, then the chances of me getting cancer increase. Specific viruses can cause specific types of cancers. We have done enough studies that we keep finding these connections between certain viruses and cancer rates, enough so that it seems like it's a real association. So if that's true, if I get a virus and then later on, 10 years, 20 years later, that virus has increased my mutation rate and I'm likely to come down with a cancer, maybe sooner than, when I'm really, really old, then that virus is involved with that cancer. So viruses are playing a role in cancers. And so we've identified a couple of viruses that we know this happens to. So there is a virus called HPV. It's called human papilloma virus, HPV. Now, if you're exposed to this virus, it's actually a really mild virus. You probably never even know that you had it. You may have some mild symptoms that you shrug off. Nobody goes to the doctor for it. It's a very mild virus. Problem is that once you've been exposed, later on, you have an increased risk of cancer. And specifically, you have an increased risk of cervical cancers. You have a 98% 98% of all cervical cancer diagnoses also have a human papillomavirus association. 95% of anal cancers are associated with this HPV infection. 70% of throat cancers, 50% of vaginal, 35% of penile cancers are all associated with this virus, which means if I can not get infected with that virus, then I have decreased my risk of getting cervical cancer 98%. Only 2% of other cervical cancers happen in other ways. And again, lots of different ways that these genes can mess up. But this is a huge deal. If I can stop my body from having that virus and feeling the effects of it, letting that virus go through until my immune system keeps it in check, I can reduce my rate of cervical cancer and anal cancer and throat cancer and those other cancers. You can reduce the rate. So how do we reduce our risk for viruses? Vaccines. Our vaccines are our best defense against viruses. So what you do is you take a little piece of the virus, inactivated, small piece, you get a vaccine, your body sees it. Now that virus is not going to hurt you because it's only a small piece. It's not able to do the damage because it's just a piece of it. But your immune system sees it. Your immune system starts to make antibodies towards it. So when you come in contact with HPV in your environment, and again, it's very, HPV infection is as high, I want to say it's like 60%. Chances are you're going to come in contact with it in your environment. I could be wrong on those numbers, but it's very prevalent in our environment. When you come in contact with it, you've got antibodies already formed, and you're going to be able to snuff it down before it does its damage, before it gets to hide and cause those, whatever it's doing to increase your cancer risk. Okay, so this makes sense, right? Let's give a virus. HPV is usually transmitted by sexual type contact, so saliva, sexual body fluids. So if we can give this virus before any of those types of contact happens, then we're protecting our kids. So that's what we've done, and that's what's come on the market. It's been called Gardasil, is the company and the name that they gave this vaccine. And so it came on the market, and they asked people, hey, let's do this, give this virus. We're going to do it before sexual activity. So if I really want to get everybody before any type of sexual activity, they're going to advertise it or recommend it for 11 to 12-year-olds at their checkups. Okay, so this all sounds great, but we just had a couple decades ago, we had that autism thing. MMR causes autism, which has been disproven. I think we talked about that in chapter 1. It's completely been disproven, but we're left with the aftermath, which is a lot of distrust on vaccinations and a lot of people who are spewing out incorrect information to push that anti-vaccination agenda, and they're worried about this. And so that's not working in our favor, the medical community's favor, the vaccine proponents' favor, because people are a little nervous about vaccines. You're also targeting a vaccine to a virus that really didn't cause a problem. Nobody knows what disease HPV causes. Like, they've never heard of it. It's not like measles or whooping cough or something that people have heard of. So you're selling it to your patients that, okay, I'm going to protect you against this virus, that if you get it, then the chances of you getting cancer 10 to 20 years from now really reduce. The problem is it is a sexually transmitted virus, so you're trying to tell the parents of an 11-year-old or a 12-year-old that you want to give them this vaccine so that when they're sexually active, which we don't want to think about our 11 and 12-year-olds being sexually active, when they're sexually active, then they'll be protected in the future when they might get cervical cancer. And it's just been a hard sell. It's just been a hard sell. So vaccination rates on that are not as high. People, again, have a hard time with it. But when we have seen vaccination, we've seen drops in these cancer rates. So, we'll see what happens. We'll continue to go through. I did the research. My kids went through that time period, and I did the research, and there were some articles out, and I was reading it. And one of them was actually sounded pretty good. And, I think that my ear is a little more trained than your average person. And they were citing peer-reviewed scientific journals, and they were saying that, that there were deaths in this vaccine trial. And it read very serious, and we all just want to protect our kids. That's all we want to do. And so so I clicked on the link, and I first clicked on the link of the site that was publishing this information. And when I clicked on the link to look at some other articles, there were some other articles that were like 4-year-old murdered by a vaccine. It was very inflammatory. So I obviously had gotten to a site that had an agenda, that vaccines were bad, murdered. When you use that kind of language, you're pushing an agenda to go through. So then, again, peer-reviewed scientific journals, I mean, they're not perfect, but they're good. So I clicked on those, and they said that there were 32 deaths. That's what the article was saying, that there were 32 deaths of people who got this vaccine. And I mean, that's serious. So I clicked on the links and I looked at the numbers. This was a huge vaccine trial, like 10,000 people were involved in this vaccine trial. They had blind groups where some groups got the vaccine, some groups got a placebo, we didn't know which, and then reported all the reactions. And again, huge numbers of people. And in the group that got the vaccine, there were some side effects reported, and there were like 32 deaths. And that's concerning, right? In the group that got the placebo, guess what? About 32 deaths. Again, the death rate, sometimes when you're talking about numbers that big, sometimes people die during that thing for unrelated reasons. If it was due to the vaccine, you would expect those numbers to be higher in the group that got the vaccine. The numbers were not. The numbers were not statistically different of deaths, mortality, which again, nobody wants to do that. Everybody wants to protect your kids. No difference in those numbers. So it couldn't be, the death rate could not be due to the vaccine. So what about some of the other things that they saw? What was due to the vaccine? Things that were statistically significant, things people experienced when they got the vaccine that they didn't experience for the people that got the placebo. When they got the vaccine, they had redness and irritation at the site of injection. They experienced pain for up to 3 to 4 days after the site of the injection. People that got the placebo didn't report that, or minor things. So that was statistically significant. People fainted. Within 15-20 minutes after getting the vaccine, there was a higher report of fainting that occurred. That was statistically significant. The people that got the placebo didn't. So again, that's probably due to something in the vaccine. Those were side effects that, given cancer, I could deal with. They felt valid. It was a huge study, well controlled. So I decided, yes, we can do that. And now, since those studies have come out, they've changed the protocols, and and my daughter actually fainted after she got the vaccine. But they've changed the protocols. They make you sit for 10 to 15 minutes to make sure you get past that thing to catch those people who can faint. Nobody wants to faint, hit your head, cause other problems. All right. So anyway, all this goes to show, do your research, but do your research on reputable sources. And this is what I'm talking about in your case studies. Do your research, yes, but make sure that you're talking about peer-reviewed scientific data, experimental scientific method type research, not anecdotal evidence, that kind of stuff. So correlation, just because the vaccine was given at the same time, just because people died in that vaccine trial does not mean that they died due to the vaccine, and that's completely different. Okay, so that's a little bit about Gardasil. Take-home message that we have found that viruses can play a role in other diseases. There's some other viruses that are coming out, like Lyme disease, causing big problems going on. So again, we're learning more and more about this. All right, last thing I want to cover, and I think this video is getting long, culturing viruses in the lab. Remember Robert Koch? That was one of Koch's postulates, that you need to culture it in a lab. Well, if you're dealing with bacteria, you could put it on agar. You can give it its nutrients, things that it needed to survive, to culture it in a lab. How do we culture viruses in the lab? We have to give it what it needs, right? What does a virus need to replicate? It needs a host, okay? So we need to give it a host, a cell that it can invade, live in. It needs a host. All right, so there's a couple different ways that we have done this in the lab. If it is a bacteriophage, and I'll go back and forth to the pictures. If it is a bacteriophage, a virus that infects bacteria, well, that's easy. We grow the bacteria in the lab, and then we infect it with the bacteriophage. What you see happening is you do a lawn growth, so we're spreading bacteria all over the cell, letting the bacteria grow on the cell, and then we're adding the bacteriophage to these healthy cell cultures. What we see, instead of colonies, you kind of see the reverse. You see whenever a bacteriophage lands, it's going to invade that bacteria and kill it. And so you see a hole, an area where the bacteria is not growing. This is defined as a viral plaque. So it's the absence of bacteria growth. And why? Because there was a bacteriophage there that got into that cell and lysed it and killed it. And so now that bacteria is not growing anymore. So this will stay. The virus is going to replicate and move through it. And then you're going to have to keep inoculating a new plate. So this bacteria, we can do plates with that. All right, but what if it's not bacteria? What if it's a human virus? Let me go back to the next slide. If it is a human virus, or an animal virus, one way that we can do this is embryonated chicken eggs. Chicken eggs is a large, they're sterile, and they have a lot of different cell tissues. So again, I keep going back to me. You don't want to see me. A lot of different cell tissues here. So if I look at that, I've got embryonic cell tissue. I've got different types of cells within that. I've got different things on the amnion. I've got all of these other things I don't even know about. There's lots of different places that could serve as hosts for different types of viruses. Again, viruses are very specific on their host, but this chicken egg allows us to put these viruses in and they can live there. And then, we can just keep them going, and do that. So we can grow viruses in chicken eggs from that. This is the reason when you go to get some of your vaccinations, you may have had the question, are you allergic to eggs? And it's not because that virus has anything to do with eggs. It's because that virus was cultured in an egg, and it's possible a protein could get swept up into the virus, into the vaccine that you're having. So that's the reason they ask you if you are allergic to eggs when you get certain vaccines, because we culture them this way. Okay, so that's one way. Another way we can culture animal viruses, providing them a place to grow. And this is a great sterile, easy way to grow stuff. What about those ones, like remember hepatitis only infects human liver cells? So to give it a host, I need a human liver. They don't let you experiment on people like that anymore. What we have done is we took human liver tumors. And so if you remember, that tumor is a growth of cells, of host cells that shouldn't be growing, and they've lost their ability to stop growing as they continue replicating. This is cancer. This is a tumor. So say you have a person that has a liver tumor. We're going to take out the liver. It's a surgical candidate. That's not take out the liver. Take out the tumor from the liver. You can take that tumor and keep it in a cell in a laboratory. And because it will continue to grow, if you give it the right nutrients, it will continue to replicate. And you can establish a cell line made up of human liver cells. Now, they're not exactly right because those proto-oncogenes, there's some mutations, but we can keep it in a lab. So for a disease like hepatitis, we can provide a human liver cell and a cell line that'll keep replicating that we can use to culture those viruses. Cell lines was a really big deal when we came up with those. It was in the 1950s. There's a book. I write this down. I know I'm talking a lot, but I love this book. It's called The Immortal Life of Henrietta Lacks. And it talks about the original cell line culture. It's a cell line called HeLa cells. And at that period in time, they were naming cell lines based on the first two letters and the first two letters of the first and last name. So this was the HeLa cell line. It was the first time they got cells to grow in vitro and continue to grow. And it's from a tumor that they took from this woman. Lots of great interest in this. It reads like a novel. It goes back and forth. Her family was poor. They were Black at a public hospital. They weren't very educated. The doctors didn't bother to explain what was happening. They created this cell line. The family didn't know, and it made a big deal. It furthered research in diabetes and a lot of diseases that we weren't able to really study because we didn't have a human host. So this was huge. But then later on, when DNA came into play in the 80s and 90s, they contacted her descendants because they wanted some DNA samples to go back and trace this, and they were related. Again, they did not explain what they were doing, and these poor people thought that their mother was still alive in a lab. Oh, it was horrible. Just the ethics involved and the racial undertones. It was a horrible thing that happened. But we learned, and nowadays you have to sign forms for everything. Can you use my cells to do this? Can you do research on this? You have lots of different types of consent forms that have to be signed, and it's due to unfortunate errors in judgment as people were working through and not understanding how this went. So anyway, it's the author is Rebecca Skloot. And after you finish this class, because it's summer and you have no time to read, that's a really good book if you're interested. Okay, I'm going to finish that up, and I think I have said everything that I want to say on the PowerPoints. We have one other thing to do, and we will finish that in a minute.